Welcome to RegenMed Global, the podcast, connecting you with the newest breakthroughs in medicine and research to help bring hope in fighting incurable diseases. My name is Shayan. Today, we're here to discuss the latest research and development of a new drug that can potentially regenerate the liver. Leading this research and speaking with us about this, we have the director of Pittsburgh Liver Research Center, Dr. Paul Monga. Dr. Monga, it's a pleasure to have you here.
Thank you.
Before we get started, would love to hear a little bit about your background.
Okay. Well, thank you for having me. It's an honor to be here talking to you about some of our new discoveries. As you said, I'm a professor of pathology and medicine here at University of Pittsburgh and University of Pittsburgh Medical Center. I'm also the founding director of Pittsburgh Liver Research Center, which is one of the 3 liver-focused digestive disease research core centers funded by NIH, NIDDK. So my background is I'm a physician, academic physician trained in India from Dayanand Medical College, Ludhiana, and then I did a lot of my postdoc training here in the United States, first at Georgetown University, then at Temple University, and since 1999 I have been at University of Pittsburgh. And one of the main reasons for me being here at Pittsburgh was a very strong and solid tradition of liver research here at the University of Pittsburgh, because the first liver transplants, the successful liver transplants, were done by Tom Starzl, and we are sitting in a building that's named after him. So there's a legacy that Pittsburgh is really proud of, and we have really built this center, and a lot of individuals are doing some very meaningful research here at Pittsburgh in the area of liver.
That's awesome. Thank you, Dr. Monga. Before we get into your research about the development of this new potential drug, do you mind maybe just telling us a little bit about what are kind of the common causes of damage to the liver, or what are some of the types of liver diseases that are somewhat common?
Sure. Yeah, so liver is, first of all, a very important organ, and we can't do without it. It performs more than 500 distinct functions almost on a daily basis. So one can imagine that you can't really be alive if liver were to give up. And what's also important to know is that liver gets most of its blood from the intestine, from the gut. Anything we eat, we swallow, it all eventually makes its way through the intestine into the liver. So it's always at risk of getting exposed to many different types of insults. So some of the very common causes of liver pathology are alcohol, and the disease that is associated with it is called the alcoholic liver disease. In the Western world now, because of a lot of high fructose corn syrup and some other artificial flavors and other types of things that are added into the food, there's also something called non-alcoholic fatty liver disease. It's an obesity-related disorder, but we have also seen fatty liver disease in lean patients who are not fat. Of course, there are viral causes of liver insult: hepatitis A, hepatitis B, hepatitis C. Hepatitis C used to be one of the biggest problems in the Western world, but thanks to a new generation of drugs that was discovered in 2012-13, HCV is pretty much cured now. So but now the focus has gone more to non-alcoholic fatty liver disease and alcoholic liver disease as the leading causes of liver pathology almost worldwide. And then there are others as well. There's autoimmune causes where the body, we don't understand the exact mechanisms, but our own immune system turns against our liver. Then there are special ducts in the liver called bile ducts, which take bile out of the liver into the intestine. There are disorders which can affect those bile ducts, and those diseases are classified as cholangiopathies. They can also lead to backflow of bile and kill the liver tissue, which can also land you in the hospital and with chronic liver disease. Other than that, drugs are a very important problem, and these are almost every drug is toxic to the liver if it's not consumed properly, including some off-the-shelf labels like Tylenol, acetaminophen that we take for headaches and stuff. Too much of it can be bad, and then there are, of course, genetic and environmental things that can also lead to liver insult. So there's a laundry list of things that can lead to a problem in the liver.
Wow. So look, thank you for sharing all that with us, Dr. Monga. So I'm curious, when there is dysfunction, ultimately, regardless of the disease that's causing it, are the ultimate result of the damage to the liver, or are they all the same, or do they differ based on disease?
That's a very, very good question. So while all the processes that can lead to liver injury are different, the pathogens could be different, the inciting agents could be different, but as you rightfully said, most of the downstream effects of that and how those patients who have liver disease manifest are really very similar. So these are going to be patients who are going to develop the most common and frequent symptom is going to be jaundice. The patients are going to come with yellowness to their skin. They're going to have yellow urine. They're going to have could have itching and pruritus on the skin. But then as the liver disease kind of progresses more, because liver performs those 500 functions, because of those 500 functions that are not going to be properly performed, the patient can come with any number of symptoms, and those can include bleeding type of disorders. Those can be, lack of appetite. People are not feeling hungry. The change of how their stool looks like will change. The urine color will change, and there can be pain associated with all of this in the right upper quadrant of your abdomen. So again, a laundry list of symptoms, but yellowness of the skin, the jaundice, those are very frequent ways that these patients can present to the clinic.
Is there a way that the liver itself can regenerate itself?
Actually, again, that's a very important question. It is very important to know that for the most part, liver has a tremendous amount of regenerative capacity. If you chronically insult a liver, it takes sometimes decades before a liver completely gives up on you. So you will see alcohol abuse, for example, which is a typical cause of a big portion of these patients. These patients would have insulted the liver through overconsumption of alcohol for many, many years, sometimes 20 years and 30 years before a liver completely decompensates and is completely replaced by scar tissue. Because again, like any other organ, eventually if you continue to insult it, it's going to get a wound healing response, just like you get cuts and bruises on your skin if you kind of fall or get hurt on the skin. If the liver is the same way, you get scar tissue, and the scar tissue continues to build up and replace the functional cells of the liver. And that's when all the other types of symptoms begin to manifest in the patient. But what liver is very resilient, as you can imagine. Anytime there is insult and injury, only a few cells die at that time, and the other cells that have escaped that damage are able to divide and replace those dying cells. And this continues to happen over many, many years. Same thing happens with non-alcoholic fatty liver disease, or NAFLD, what we call. Because of metabolic stress, because of genetic susceptibility, part of your liver, part of your cells are dying off, but the other cells are continuing to replace them. But over many years, the cycles of insult continue to progress, and the functioning mass of the liver eventually is decompensated. So you are not able to then proliferate and or divide as much. And there is also inflammation. A lot of immune cells come in, and that can cause a lot of problem. Can result in cancer at the end of the day. Can result in rapid liver failure, what we call as acute on chronic liver failure. So, but it can take many, many years. So, as you rightfully said, liver has a tremendous ability to regenerate, and it takes years of insult. The only exception to that rule is if you have an acute insult or acute injury to the liver, and that usually is drug induced, as you can imagine. Or if you are an alcoholic drink, and if you just consume way too much alcohol, then you can develop, a certain type of injury, alcoholic hepatitis or drug-induced liver injury, what we call as DILI, and that can cause a very acute failure of the liver. That will happen if you, for example, if it's a suicide attempt by somebody or accidental consumption of certain drug, which can result in acute. But for the most part, your liver will continue to grow in the face of an insult, provided there are sufficient number of cells, alive cells, that are left in the liver. And so it can usually take sometimes 2 decades or 3 decades before the liver will completely fail.
What's the current protocol of being able to treat the damage or being able to stop it from degeneration? Can you tell us a little about that?
Absolutely. As you can imagine, it all depends on what the primary insult to the liver is, right? So again, we can talk all day about the different causes and how those are treated, but in the big picture, if you can stop the insult, and those are sometimes not as trivial as it sounds. If a person is overweight, losing weight can be a challenge. So it's not just say, oh, you have to eat healthy. It's easy to say that, but there is a strict discipline that comes with eating healthy, with exercising on a regular basis, and that requires a certain mindset. So a lot of good academic centers will now have multidisciplinary teams where a person who's overweight, for example, would work with a nutritionist, a dietitian, will work with a hepatologist, would work with even a psychologist and a psychiatrist and an endocrinologist, and then this team of individuals will work. And if you are able to kind of allow this patient or stimulate this patient to do all the necessary things like behavior modification, weight loss, you can really stop the disease process and even reverse the disease process. The same with alcohol. If you completely undergo abstinence, it could be because of psychiatric intervention, could be because of other interventions, and those can really reverse. For viral hepatitis, luckily we have very strong and important advancement in the field has been the drugs for hepatitis C virus. Hepatitis B, we have very good vaccines, but those people who refuse to get vaccinated for hepatitis B will be always susceptible to getting hepatitis B. And then once that progresses, it can cause its side effects. Unfortunately, there are a group of diseases for which we don't have much cure. For example, the cholangiopathies that I was talking about, the bile duct disorders, they are called primary biliary cholangitis or primary sclerosing cholangitis, PBC and PSC. Those are black box of hepatology. We really don't understand what is the mechanism of why the bile ducts begin to get destroyed in these, and those patients, unfortunately, there are very limited drugs that are available for them, and hence these patients oftentimes will end up needing a transplant, if they have to survive. And then there are other pathologies like that, autoimmune hepatitis, certain group of patients. So we don't have cure for a lot of these pathologies. And if these pathologies continue to progress, they all result in eventually cirrhosis or end-stage liver disease, liver failure, which would require a transplant. But again, as you know, transplants are not available. We don't have liver for everyone, and hence, we have to be creative in how we can help these patients. And that's where some of the work that we and others are doing has been relevant, and hopefully going forward we can have a dramatic impact on the landscape of liver pathologies.
That's some very valuable information. Thank you, Dr. Monga. I'd like to now get into your research. You and your team developed an antibody that can potentially regenerate the liver. I mean, that's some very exciting things. Can you tell us a little bit about what prompted you to develop something like this?
Right. So I'll maybe make a little correction here. So we were not the ones who developed this antibody. This antibody was developed by good friends and collaborators at a company called Antlera, which is actually San Francisco based, but the original individuals who have discovered this technology are from University of Toronto.
Okay.
And we have collaborated with them. Now, the reason how this collaboration sort of worked out was because I have a long-standing history of working in the area of liver regeneration field. We are interested in how a normal liver, when you remove 2/3 of that liver out, the remaining 1/3 of the liver has the capacity to grow back to the same mass, to the same functional capacity as the old liver. So we have been really interested to kind of understand which signaling pathways, which proteins are making the liver grow back to that normal size that is required by that animal to survive. And that's where we discovered the role of Wnt signaling pathway. And I won't bore you with the details of this pathway, but this particular protein, we have really over the last 22 years, my lab has been really at the forefront in discovering the exact, what I call the cell molecule circuitry of the liver regeneration process. And Wnt's are very important. So and again, in order for you to be able to appreciate, or the listeners to be able to appreciate how this antibody works, it's important to know that these proteins called Wnt's are released from a special cell in the liver called endothelial cell. And then these Wnt's then bind to their receptors on liver cells, which are called hepatocytes. Hepatocytes are the functional unit of liver. These are the cells that perform the 500 functions that I was talking about. So endothelial cells release Wnt's that bind to the receptor frizzled and co-receptor called LRP56 that then leads to beta-catenin translocating from the cytoplasm into the nucleus of a liver cell, where it then activates cell division program through regulation of cyclin D1 and other cyclins. So that cascade that begins right after a part of the liver is taken out, that cascade begins in the remnant liver, whatever liver is left in a patient, in a mouse, in a rat. That's the cascade that begins. So once that cascade begins, there is process of repair that starts, and within 24 hours in a rat, 48 hours in a mouse, you begin to see liver cells are now beginning to start to divide. And it's a very short process. Within 24 additional hours, the liver had already, all the liver cells, hepatocytes, have divided, and then half of them divide one more time, and then it takes about 7 days in a rat and about 14 days in a mouse for the liver to come back to the same size, and then it stops. So our question was, we know that Wnt's are playing very important role in beginning this cascade. What if we can activate this pathway? using so that it doesn't have to depend on the winds coming from endothelial cells. So that's where this antibody that was developed by Antlera became very, very meaningful to us. So this antibody recognizes the receptors that are present on the liver cells, goes and binds to them, and then activates beta-catenin and translocates, helps it move into the nucleus where it can start the cascade. But remember, it can do that without any exogenous winds, without any endothelial cell winds. So that was, what was very exciting to us. So when we took a normal animal that had not gotten partial hepatectomy, where we had not performed surgery, and just gave this antibody to them, the liver started to proliferate and grow, which was very exciting to us because we were looking for ways to kind of improve the sort of the regeneration of the liver or the size of the liver. And so that was sort of the first way, first sort of experiment that we did. And then we decided to do a little bit more translational experiment, and that's where we challenged these animals to Tylenol. As I said, Tylenol overdose is a major cause of acute liver failure in the Western world. It can be suicidal, can be because of accidental overdose, especially in pediatric patients, or children who don't really know what they are being given, but the parents are giving them some cough suppressants and some Tylenol together, not realizing that some of these cough suppressants also have Tylenol. And then because the kid is not doing well, they continue to give more and more, and eventually the liver of these kids kind of fails. But in all, so we decided to kind of create that model in mice where we gave a high dose of Tylenol into this mouse, and then within about 24 to 28 hours after, when the liver is beginning to fail, we give them this antibody to activate the beta-catenin pathway, so we can promote the repair process. And lo and behold, when we did that, we saw that these animals had much better liver regeneration. Their liver function tests, which are from the blood that we draw, we can look at the liver function. They were all significantly better. And when you look at the slides obtained from these animals' livers to see how the repair is happening under the microscope, you can see the liver is looking so much better, healthier, and almost restored when you were giving this drug. So it was very exciting and very promising for all of us, and very gratifying for all of us to see. So that was in just the entire sort of excitement with this particular agent.
How long did the improvements continue for?
Yeah, so one has to realize that the models that we can test these drugs all have their own sort of strengths and weaknesses, right? So the model that we use is an acetaminophen overdose model. When you give over this particular drug, so the liver damage begins to be visible within 6 to 12 hours. By 24 hours, it's really bad, and then the animals will, within the next 12 to 24 hours, begin to die, or they will begin to spontaneously recover. That's how this animal model works. But when we did that, and we started giving this drug, none of the animals died. And all of these animals, when we looked under the microscope or looked at their blood reports, they were significantly better, and these animals were healthier. So, we just gave this drug once, and then we were able to show this. So in an acute liver setting, in patients, I would imagine a very similar. If you have a patient who comes in to the clinic with acute liver failure because of drugs like acetaminophen, after very careful assessment, and I can go into more detail a little bit later on why I'm saying very careful, time frame that needs to be considered, one can give this drug to promote the regeneration and help either prevent transplant or at least bridge the patient to transplant until a liver becomes available, etc.
With these chronic diseases, is there a way to be able to avoid a liver transplant by using an antibody like this?
Yes, so that is again a very, very relevant question, and I have been asked that question many times. And the short answer to that question is I don't know. And the reason for that is we haven't tested this in a chronic setting. But going forward, this is one of the key things. I have graduate students and postdocs in the lab who are really interested to pursue this. So we have at least one graduate student right now who's working on that. We are now, unfortunately, I'm not going to go into the details, but these animals can have a 30-day disease period, and after that they die. So it's a little bit more chronic. It's not like acetaminophen injury where everything happens within 48 hours. So these animals will have about 30 days, although there is variability. So we are starting to give these animals this particular drug, and it will take us another 2 to 3 weeks before we know whether this drug would be effective in that. But then we also have other models which can be about 3 to 6 months long to develop the kind of disorder and disease and will eventually result in a pretty bad pathology of the liver. We haven't tested this drug in that system yet, and just because it's not easy to get this drug. They are collaborators, but again, they have their own priorities. They have been extremely helpful, highly collaborative. But again, until they are ready to take this to the next level, and so you have to be very creative in identifying where do you want to use this first, what are the proof of concept studies that are required in that space. So we are going to be working with them later this year to kind of map that out and then decide on some of these chronic models where we can use, including fatty liver disease. And we can create fatty liver disease models in mice and rats, although they are not as severe as humans. But that can be done, and a couple of other different types of models. And it'll be very interesting to see if this antibody, which can activate WNTs, can have a positive impact on resolution of the disease.
So with all the different areas that you're looking at, when you described acute, chronic, and so forth, how long do you see these studies going until you guys have an understanding within these animal models?
So I think the best way to answer that question is that I think we are fairly confident that this particular drug could benefit patients in acute setting almost immediately. I think the only thing that is left to do is proof of concept studies in patients. Of course, even before that, we have to do safety studies, or the company will have to do safety studies in healthy population, which is called you have to file for something called IND with FDA, investigational new drug. Then you have to do phase 1 clinical trial, which is for safety, etc. And once these drugs are not having any side effects in patients, only then can you decide what your clinical trial is going to look like. And I think these, in my mind, there are at least 2 or 3 different types of settings where these drugs could be used right away because they require very short administration. One is this drug-induced liver injury, which is in acute form, like acetaminophen overdose. The second would be in liver transplantation setting, where a lot of patients are being transplanted. And, if this drug can be given almost prophylactically to, stimulate liver regeneration right after a patient has been transplanted to see how quickly the liver functions can return to normal if you were to give this drug, and how much that can help with hospital stay. If you can cut down the hospital stay of these patients, help them regain their strength, regain their liver function sooner, that would be, I think, a valuable subgroup of patients. And then the third one is something called small for size syndrome, where the livers are smaller for the size of the patient after transplantation. The graft that was available is smaller or is not growing to the same extent as was expected. In those cases, this drug could be very useful to kind of test its ability. So I think those would be some of the acute sort of indications that I would definitely say would be the low-hanging fruits. And then comes, the harder question, which is what about chronic liver diseases, which are the big health burden around the globe. And I think that's where we'll have to identify the right subgroup of patients which would value. And I don't think we have enough preclinical data at this point in time. And I think that's something that I need to address in my lab. And our goal is that by the end of this year, based on our conversations with this company, we are going to start examining those specific situations for which animal models are good representatives of human diseases, and then test these drugs out in those pathologies in my lab or in the labs of other collaborators that this company may decide to identify. And then all of us work together to really work towards that goal of treating all these varying eclectic pathologies of the liver.
So knowing that the type of effect it could have in the right group that could benefit from this, how soon, and I know there's still a lot of research done, but how soon could something like this be available for humans?
So I would say that with the current pace that we are going at, and again, playing a little bit on the being a little bit more cautious, I could see if the company is able to, of course, raise the funds that are needed, because the bottlenecks are, experimental, but the bottlenecks are also logistical. If everything were to go smoothly, I would say in 2025, 2026, one can have, phase 1 clinical trials to address the safety issues, and then, it will take another, couple of years, after that. So well before 2030, I can really see, even clinical trials being read out in this space for limited number of liver pathologies. And that would be the goal, and that should be what we should all be focusing on, those who believe in regeneration-based therapies for the liver.
Could the development of a drug like this, could this open up opportunities to treat regeneration of any other parts of the body or any other function of the body?
Yeah, that's a great question. Yeah, the Wnt signaling is not just unique to the liver. What is unique to the liver is its capacity to regenerate. There's no other organ in the body that you can cut in half and the other half, can grow back to its own size. There is just no. So for whatever reason, liver is, bestowed with that sort of capacity. But those are the lessons that we can learn from the liver. Those are the pathways that we can learn from the liver, and then apply it to different organs. So Wnt signaling is very important in our skin renewal, in our intestine renewal, because there are stem cells in skin and in gut, hematopoiesis, the blood formation. These pathways are very important in that. Now, could a Wnt agonist be used to repair some of those conditions? Like, for example, a big pathology, which again is an unmet clinical need, are inflammatory bowel diseases, IBD, where you have Crohn's disease, you have ulcerative colitis. These are conditions where the intestines are beginning to not their stem cell compartments are defective. There are a lot of ulcers that begin to form within the gut. So can this drug be given to promote repair in that in that context? And the answer is, theoretical answer is yes. And I know there are studies ongoing in some of the key labs where they are using organoid cultures from patients, from human cells and animal models of gut injury, where they are trying to use this drug to induce repair. So I think there's going to be a lot of promise of inducing repair using regenerative therapies such as this beyond liver.
Excellent. Going on the topic of regenerative medicine, I know something that's been popular, something that at least research has been coming out, is about the pretty much about stem cells being used for liver disease, liver damage, liver regeneration. I was curious to see if you have any thoughts about the effects of stem cell therapy on liver regeneration and how that might compare to the antibody.
Right. I think the first thing I should just, as a cautionary tale, I should say is that when we say stem cell therapies and when we say, we have to be very careful. A stem cell has a proper definition. From a scientific perspective and from a physician perspective, stem cells are a very small group of cells which you have to check mark so many things in order for a cell to be called a stem cell. So a true stem cell therapy, from a scientific and a clinical standpoint, is very limited in its scope at the present time. You will hear all these other stories come to this country for stem cell therapies and for curing cancer and this and that. Those are, again, one has to be very careful as to what they read, and you have to talk to a professional, a physician and a true scientist in order to be able to kind of clarify what it truly is stem cell therapy. So when you talk about really a stem cell therapy, there's very limited stem cell therapy that is FDA approved and given in the clinic, and the only one that comes to my mind is hematopoietic stem cells. And that is, in the setting of cancer, because you are ablating, the cancer cells, but before that you are collecting stem cells from the same patient. And then once you kill the cancer cells with chemotherapy or whole body radiation or other regimens, you then transplant the stem cells that have been collected from that patient back into them so that you can now recover their hematopoietic system. So that's pretty much, the scope of stem cell therapy, unless I'm grossly missing something at the present time. But that's what stem cell therapy is. Now, you see a lot of preclinical studies in animal models where people have used stem cells from different organs, or to replenish, and repair pathologies. And that is, again, all in preclinical space, in mouse models, in rat models, in zebrafish. There are studies that are proving that concept. From a liver standpoint, I can tell you, in my experience over 25 years of me being in this area, I don't believe much in stem cell therapies for the liver. And the reason I say that is, liver is unique in so many ways. Liver, as you mentioned at the beginning, is one of those organs which knows how to regenerate and repair itself. So there are so many different mechanisms that liver possesses. That is why it can take up to 20, 30 years of chronic insult for a patient to come in with a liver failure. But it's and again, I don't want to bore people with details on what those different processes are. Suffice to say that that if one level of repair fails in the liver, there is a quick thinking on the part of the liver, and it will go to the second tier of repair. I call it tiers of repair. And there are many tiers of repair. If the first tier fails, then liver automatically defaults to second tier of repair, then the third tier of repair, then the fourth tier of repair. So the stem cells, if they exist in the liver, and that's been a very contentious issue, if there are two true stem cells that check all those boxes that I talked about, then that may be the fifth or sixth tier of repair in the liver. Usually you don't have to get to that point, you will be able to kind of figure out a way to repair, before that. But when ultimately all these tiers fail, that's when patient is going to run out of options and will come to the clinic seeking help, seeking what can be done to kind of to fix them. And I think our focus should be more on figuring out what is the best way to treat fatty liver disease, what is the best way to treat alcoholic liver disease, cholangiopathies, autoimmune hepatitis, because those are still challenges to treat. There aren't any medications that you can give to stop or reverse, unlike some of these viral hepatitis, where we know it's an infection. And we know, just like antibiotics, we have antivirals that can be used to kind of treat them. But again, those treatments are not completely optimal, but it's better than before, and I think they'll continue to get better over the next several years.
Dr. Monga, you've educated me so much about this topic, and I'm sure the viewers themselves would also appreciate the type of information that you're providing. I know we're getting kind of at the top of the hour. I'm curious if you have any other kind of final thoughts that you'd like to share with us about the development of this recent drug or anything of that nature.
Yeah, no, I think I would just say that, in this world, where, I came from India, and a lot of the issues associated, at least the time when I was growing up, when I was going to medical school, a lot of that was associated, the pathologies were associated with undernutrition, malnutrition, when there was not sufficient, materials going inside the body. But things have changed, even in India, which has progressed, and many other places which have progressed, and we are now adopting more and more Western lifestyle. And here, overnutrition has become a big problem. We are not taking care of our bodies. We are not taking care of our nutrition, what we eat, what we drink, and I think a lot of the things that we have talked about today, a lot of them can be prevented or can be slowed in the process. So, my final thoughts is medication should always be secondary or tertiary in your mind. If you can prevent, if you can somehow adopt a more healthy lifestyle. I know it's easier said than done. I often tell that to myself, and you have to lead by example if you have to kind of say these things to others. And it's not easy, to be very dedicated in what you do, what you eat, how you exercise, and our lifestyles are very sedentary. We have talked about, this sitting on a chair for an hour, right? And I'm going to have to be in a meeting for another hour, and I'm going to be sitting on this chair. So our lifestyles, just require us to have very limited mobility. If we can change that, if we can incorporate some more, regimented exercises in our lifestyle, and at the same time we eat healthy, we drink everything in moderation, and if we can prevent something, we prevent that. That would be, I think, where we should be focusing on. But once those things begin to kind of progress, there are genetic components to this, which we are unaware of, but we are finding more and more. There are certain polymorphisms in your genes which make your liver worse off when it comes to fatty liver disease. So if you are just kind of dealt with those cards, there's very limited things. If other person has to insult their liver, a certain number of times, if you have those genetic vulnerabilities, even half those insults will be enough to disease your liver more. So I think those are the pathologies those are things that are out of your control. But again, I think focusing more on preventive lifestyle changes in your lifestyle, behavior modification, if you can help it, is the best way to go. But after that, I hope that the discoveries that we make and others in hepatology are making will eventually help our patients who need this urgently, in my opinion.
Those are some great final words, Dr. Monga. Thank you so much. It was truly a pleasure to go and be able to to listen to what you have to say, and just some groundbreaking things that you and your team are leading within the field of medicine and research. So it was a pleasure. Thank you so much, Dr. Monga.
Of course, Cheyenne. Good luck to you, and keep on doing all the important work that you do. Take care.
Thank you.
Bye-bye.
You too.